So I'm Daniela Marazziti, and I work in Monterotondo in the group of Glauco. And along with the big project on knockout production, phenotyping, and archiving, in Monterotondo we also would deal with small, more human projects that maybe is what you can do when you go back to your lab. So we were also alive in the genomic era, and in that era we cloned 2 genes. And from that time we remained stuck with these genes, and our project is to find their function. This gene, they have this horrible name, but they have an ontologic meaning. And this name, they stand for G protein couple receptor, because number 37, because this was the 37 orphan receptor that was cloned. And its closest homologue is named GPR37, like protein 1, because they constitute a small subgroup belonging to this big, enormous gene family of G protein couple receptor. What I would like to discuss at the end is whether there is any advantage in being a knockout or not. And then I think this is quite interesting, and we will decide at the end of the talk. It's nice that Andres spoke before me, because I will speak a lot again about cerebellum. So you will be convinced that it's such an important area in the brain. So the 2 genes belong to this G protein couple receptor or 7 transmembrane receptor. And even if you don't work with them, now you realize they're very important, because each of the speakers, when they speak about their work, were mentioning this family. This family constitutes by far the most enormous protein family. And the members of the family are evolutionary, quite triumphant, because with the evolutionary time, they acquire the ability to transduce almost everything. They can transduce light, ions, hormones, peptides. I mean, they are the only sensor that a cell has to understand what is going on around. And as you know, this is very important for cell communication and functioning. This family is so important that almost 60% of the drugs actually on the market are against those. And last year, the 2 big players in this field, they were the first one who cloned the first member of this family, received the Nobel Prize. So there are some things that we know about our genes and something that we discover with the time. Now it's nearly 15 years we're working with this, so we know every day more. So the ligand for both genes, 37 and L1, is unknown, and so also the G protein. Though in some cell types, it seems that they are linked to a GI, an inhibitory G protein. What we know, there is something we know, is the sequence, the chromosomal location, and the expression. And this is what we started with. We cloned the gene in human, in mouse. Somebody else did it in the rat. And the chromosomal location of both genes, and this is interesting because these are in syntenic region because human and mouse, and this means that these genes are very important, because when you have conservation also in the chromosomal location, this means that it's important to have it in a proper location. And then we have the expression that I will speak a lot about, and you will see. All this we do because we want to know which is their function. Like you know, I mean, now we have to put many things together to have, at the end, a single output. And now I built up the puzzle, but I can tell you we're just at the beginning, even if we had more and more data, but we haven't filled up the picture completely. So we start from the gene and the protein, because from the structure, we know that they belong to the G protein couple receptor. We know that they have 2 exons, 1 intron, so if we want to produce the knockout, we know where we have to target. We know that they are homologous to peptide receptor, So for sure, they will have to bind a peptide. So we did extensive search for peptide, but we didn't succeed. The pharmaceutical didn't succeed, and so we are still stuck with that. Then we did a lot of the expression, because this we needed to, in the case we want to produce a knockout, as we did, where we have to look and which is the developmental stages that could be interesting. Then we produced the knockout using the Krylog strategy. I will not show it to you because it's quite banal. And we are working, all the data I will present are with the full knockout, the traditional knockout, but in the freeze we have the floxetal. And I think in some cases maybe it would be better to go back and look also for the conditional knockout. And then for many, many years, we went through a lot of phenotyping characterization, and now it's clear to you. Now we have to start with the anatomy, histology, as Paul was mentioning yesterday, and then after that we perform a lot of biochemistry and behavior. Luckily, in Monterotondo, we could do all that because we have all the facility to do all the experiment. So I will introduce you the cerebellum because I want to speak to you at the beginning with the first knockout. That is the second one that we produce. So it's the L1 protein. It's the GPR37-like protein 1. And this gene is specifically expressed only in the cerebellum and only by the glial cell of the cerebellum that, as you will see, they are quite peculiar and special. But before I go into detail, I just want you to go back to remember what is the cerebellum. So the cerebellum is this red structure here. It's mainly composed by a central structure that is named vermis and by two lateral hemispheres. It is very important because it is involved in several functions that has to deal with movement, but not initiation of movement. It's just movement coordination. So when you have defect in the cerebellum, you are able to walk. So you can start walking, but you move strangely. You don't have coordination. And then this has to do with muscle tone and also motor learning, because the cerebellum is integrating a lot of data coming from the spinal cord, the brainstem, and the motor cortex of the brain. If you section the cerebellum at the level of the vermis, and all the data I will present, they come from the vermis, so the central part of the cerebellum, it has this structure that you have already seen with André. As you can see, it is characterized by fixture and lobes. And if you go at the cellular level, I only will speak about the measured cellular type, but doesn't mean that the others are not there. It's just for simplicity. You follow easier. There are two major neuronal populations, the granule cells and the Purkinje cells, and these Bergmann glia cells, the red ones, that are astrocytes, but they don't look like a star, like normally happen with astrocytes when they are mature. The Bergmann glia cells, they keep this sort of precursor-like structure with the cell body and long fibrous filament. They are not organized randomly in the cerebral cortex, but they are organized in layers. So the granule cells are in the more deep layers. The Purkinje cell body and the Bergmann glia cell body, they lie in the so-called Purkinje cell layers. And then above them, you have the molecular layers where there are only few cell bodies, but mainly the dendritic arborization that is huge, as you have seen with André, and also the filament that they come from the Bergmann glia cells. The granule cells and the Purkinje cells are very important for the brain functioning because they provide the cerebellum output. But the Bergmann glia cell role is not very high investigated. So everybody claims, oh, in adult cerebellum, these cells will provide nutrient to the Bergmann glia cells. That is the only neuron that provides information to the brain. And during development, because it has this strange structure, it will provide support where the granule cells could move. Because if we go during postnatal development, we have to pass from a sort of globular structure to this highly sophisticated architecture in the cerebellum. The development of the cerebellum occurs in many species postnatally. In the human, it takes 2 years. In the mouse, only 25 days. And in these 25 days, you have to do a lot. And mainly, you have to move these cells here that are represented in green from this location to the inside part of the cerebellum. And while the Purkinje cells and the Bergmann glia cells that here are represented as dots are already in the right position. So very schematically, what happened in the postnatal, in these 25 days after birth, is that the layers are different from the adult cerebellum because now we have 4 layers. In addition to the molecular layer, we have a more top layer, and this layer is called external granule layer. And at birth, the precursor of the granule cells are already here, and in this, the first 3 weeks, they proliferate quite actively. Then they move inwards, and they move in the so-called inner external granule layer. And here, they become postmitotic, and they start to differentiate, producing this prolongament. After differentiation, they continue while they move, they start the migration. And these are the steps that in time they take different times. So this process will take something like 40 hours. The migration is very fast, it's only 4 hours. And during the migration, they produce that are the parallel fibers that are very important for the wiring of the cerebellar Purkinje cells, as you will see later. So they move, they extend their filament, they produce the parallel fiber, and then finally they reach the internal granule layer. The expansion, the postnatal expansion of the granule cells is enormous because they are the thinnest neuron in the brain, but they are the most numerous. They constitute 3/4 of all the neurons of the brain. So you can imagine how important they are. In the Purkinje cell layer, there are already positioned the Purkinje cells, but what they do in these 25 days, they maturate. And I will show you later a picture that is more clear. And in the same position, very tightly bound to the Bergmann glia cells, to the Purkinje cells, we have the Bergmann glia cells. With their structure, they provide a tracking system for the movement of the granular cell. So the granular cells, they proliferate, they differentiate, they migrate, and then they maturate when they are in the internal granule layer. And in the meantime, the Purkinje cell layer, they do a lot of changes. So this is the situation if you stain a single cell body at P0. So the cell body is increasing in size, but what is fantastic, I think, is the enormous dendritic arborization of these cells. This is the only output of the cerebellum. If you read in the paper, everybody claims also the Bergmann glia cell maturate. And there are specific markers for maturation or for non-maturation, like nestin, that is a marker of non-mature Bergmann glia cells. There are some markers of mature Bergmann glia cell, but really nothing is known about that. They are just there to support the function, but this is not our idea. So recapitulating, in the postnatal cerebellum, you have 4 layers. In the adult cerebellum, only 3, because the external granule layer, once the granule cells have proliferated, they move out, so that layer tends to disappear. And in the meantime, you have this huge proliferation of the dendritic arborization of the Purkinje cells. Luckily, as so many things happen, there are many genes that are up-regulating and down-regulating. And so you have specific markers for almost everything. And this is important because if you want to understand what is going on, you have to follow proliferation, migration, maturation, and so on. And so this is what is going on. At the beginning, essentially, you have to sustain proliferation. So you have the activation of cell cycle-related genes. Then the cytoskeleton is important because the granule cells has to turn, the body size has to move, and so cytoskeleton is very important. And later you have to provide myelin and synaptic wiring to the Purkinje cells, so you activate these genes. In fact, during these 25 days, also the synaptic wiring of the Purkinje cell occur, and in this period, the Purkinje cell make contact with the parallel fiber, so each Purkinje cell is contacted by almost 200,000 parallel fiber per Purkinje cells, while it is also contacted by the so-called climbing fibers that are not produced locally in the cerebellum, but they come from the more deep cerebellar nuclei in the inferior olivary system. What happened from P3 to P15 is that at the beginning, there are more than 2 climbing fibers for Purkinje cell. During the maturation, the climbing fiber has to be reduced to 1. And this occurred after P10, before P15, so in a very limited time. Only 1 climbing fiber is selected, and this rope completely around the cell body and the dendritic arborization of the Bergmann glia, of the Purkinje cells. This is very important because the rod-like structure is the base of the rod-like structure of the Bergmann glia cells is the point where the Purkinje cell dendrite attach, and they are sort of track high toward the apical of the cerebellum. So you understand that in our mutant, where we have a reduced presence of the tubular structure of the Bergmann glia cells, it's clear that the Purkinje cell dendritic arborization is more expansive. Could be just a physical problem. So now here we have a strange phenotype because we have a receptor that is expressed in Bergmann glia cells, and these affect granule cell proliferation, not the migration, Purkinje cell maturation, and also Bergmann glia cell maturation, as I think I showed to you. So we have to deal with proliferation. Again, we have many choices. We have to go through all these different pathways. And luckily, at the end, we found that there was something going on with this pathway, the Sony-Hedgehog pathway, that is very common in any part of the body when you want to have cell proliferation, tumorigenesis, and stem cell maintenance. If we go back to the first question we asked, which is their function, we still don't know, but we have some hints. So we know that if we look at postnatal cerebellum, GPR37L1 interact with patch, regulating granule cell proliferation, Bergmann glia, and Purkinje neuron maturation. And so from what I will show you also in the next slide, we conclude that maybe it's better not to have this receptor, because when we put our adult mice again in the rotarod, because this is still the best test you can have for motor balance and coordination. And we perform this experiment at different ages in adult animal. What we have with different trials is that our knockout mice are always performing very, very better compared to the wild type. And if you put all the different trials together, what you notice is that with age, the mice, they tend to lose coordination. This is normal. But our knockout mice, they remain very, very active. They seem to be better coordinating. And this probably has to do with the better maturation of the Purkinje cell early in the postnatal development. For sure, now we will have to perform physiological data, because in the postnatal development is the period where long-term depression is activated. And P20 is the day in which long-term depression starts to be visible. We are pretty sure that in our knockout, we will have long-term depression activated before. So this is everything regarding GPR37L1. The other gene, I will go very quickly through, but I don't want to confuse you. Okay, clearly we will have to study the role in ciliopathies and in medulloblastoma, because we have overexpression of patched, overexpression of anemic, and they have to deal with medulloblastoma. But this I hope I will present the data next year. And now we go to the other one. The other one, they are very close homologs. So we reason that they will have the same function. They will bind the same ligand. But what the second one has, and this is something you have to remember, it has this long N-terminus. And although the genomic sequence have been done, we don't find anything in the databases that looks like this long amino terminus. The expression is different from the one of GPR37L1 because it's almost everywhere in the brain. So now we know that GPR37 is expressed by the other family of glia cells, the oligodendrocytes, but it's also expressed by a tiny population of very important neurons, the dopaminergic neurons that are the ones that degenerate in Parkinson's disease. What is important to remember is that these two receptors, they differ only for the amino terminus, but when we try to express GPR37 in cell culture, any cell culture die. I mean, we spent years not understanding what was going on because we were creating stable cell line, we were defreeze them, and there was nothing. Luckily, we understood that this receptor, it has a high potential of aggregation. So if you tend to overproduce the receptor, it gets accumulated in the endoplasmic reticulum and any cell die. A Japanese group overexpressed GPR37 in Drosophila cells, and that is very nice because there are only 8 dopaminergic neurons. And if you overexpress GPR37, the 8 dopaminergic neurons are gone. So there is a sort of specific toxicity for the dopaminergic neurons. I don't have the time to show you why, but it's important to remember that it's important, so important that GPR37 aggregates are removed from the cell, because the cell will die, that there are 2 ways in which GPR37 could be removed. One is via the proteasome, and the other one is via the autophagosomes. And we have, if you look in the literature, you will find the data. clearly, as the dopaminergic neurons die, GPR37 is involved with Parkinson. This is all published. You can have a look. And is found in the specific aggregates that are typical of the Parkinson disease that are the Lewy body. This is GPR37 accumulated in the Lewy body. And the knockout we produce has interesting phenotype because it has reduced weight, reduced dopamine content, and reduced locomotor activity and motor coordination, as you can expect. I want to reach this point. What we found out is that GPR37 co-localized with the dopamine transporter. That is very important for the dopamine cycling around the dopaminergic synapses. The dopamine transporter, I will skip all this, is very interesting because now we go back to patch. The dopamine transporter and patch are structurally similar. So GPR37 and GPR37L1, although they are expressed in different cells, they do the same job. They interact with this type of molecule, the transporter-like molecules. And all our study in the past with this GPR37 has to deal with the dopaminergic neurons. But I told you that this is also expressed in the oligodendrocyte. And Sony Hedgehog regulate also oligodendrocyte maturation. We have started a project that will deal also with this. So these two receptors was true that they were highly similar and they will do similar function. And this is what we found out with our study. Also in this case, it's better to live without GPR37 because although you have some motor defect, your neurons are more resistant to dopaminergic specific toxicity. You can read this in our paper. I want to thank the people who did the job. I have to say that Federico, that was the former student of André before, was working with us, and he participated in the construction of the first knockout. And this is the people who did the job. We are quite a good group of people because we have different expertise. So we have people like Silvia and Elisabetta that are experts in behavior, while me, Chiara, Gina, and Raffaele, we are more experts in the biochemical part and in the anatomical part. Then all this was possible because we have collaboration with the people next door in the MBL outstation, and also thanks to the people from the EMA archive who help us a lot with the cell line. I want to thank you, and I'll show you also some pictures because there are some beauty in science. And this is from Cerebellum. Thank you.